So this is a cross section of an insect. It's a grasshopper. So when we cut it open, this is these are all the structures that are important in producing hormones in insects. Right? You can see the brain here on top of the head itself, and then it is then connected to other organs here or other cells. So we call it as corpora alata and corpora cardiaca. And also we're going to look into prothoracic glands, which is the one that is producing the steroid hormone. And then we're also going to look into ventral ganglia, also producing hormone. Okay, so we're going to look into that. So this is ventral nerve cord. So in brain, we have neurosecretory cells that are probably producing neuropeptide hormone that can control the hormones that are being released in prothoracic gland and also in corpora alata and corpora cardiaca. Right, so you can see here we have corpora cardiaca, and just next to the corpora cardiaca, this one here, two structure here, is what we call as corpora alata. Right, so we're going to look into the structure itself. What does it do? Right. So corpora alata, the normal secretory function is ensured only when the integrity of the central nervous system, the corpus allotum complex, is unbroken, and the timing of the activity of the corpus allotum is controlled by the brain. What does it mean? It means that this corpora alata is directly connected to the brain. So it can only function if the connection is unbroken. There is no obstruction in terms of the connection between corpora alata and the brain, and they are connected by blood vessel, right? And corpora alata is also controlled by the brain hormone. So corpora alata, the normal regulation of the corpus allotum is dependent on its connection with the central nervous system or the CNS, and the signal to the corpus allotum to change its secretory activity comes from the brain. So insect endocrinology provides some of the paradigm that are used in the studies on the regulation of juvenile hormone biosynthesis. So we have corpora alata here, and corpora alata is the one that is juvenile hormone. Later on, we're going to look into why is it called a juvenile hormone. So how is the regulation of the juvenile hormone? And juvenile hormone regulation is basically controlled by the nervous system itself, the brain. So juvenile hormone is secreted by the corpora alata prior to each moult. So JH secretion by the corpus allotum is regulated by two neurohormones produced by the brain known as allotropism and allostatin. So if you can see, remember your, I think, growth hormone, we have somatostatin, somatotropin. So it's the same function here. All right. So juvenile hormone is released by the corpora alata, is needed when in the life cycles of the animal. It basically drives the molting process in insects. All right, it basically drives the molting process in insects. Molting is basically the changing or the growth of insects. All right, in every in life cycle. Later on, we're going to look into that, and we're going to look at the level of juvenile hormone and how it basically plays its role in the development of an insect. And these juvenile hormones, they are basically controlled by neurohormones. So we call it as allotropism is the one that stimulates the production of juvenile hormone, and we have allostatin. So basically, it inhibits the synthesis of juvenile hormone. So it comes from the word statin here. It becomes static when JH hormone. It becomes static when allostatin is being released. And this is basically the placement or the location of corpora alata in different insects. So we have A is basically in mosquito, right? Just behind the brain, corpora cardiaca. And then we have corpora alata here. And then we have B is a cockroach, and C is a hemipteran, right? these are all insects, but different insects, and they have different what we call is different, the same location, right? Just with the connection between the brain and corpora alata, but different in terms of the size itself, all right? But the function is still the same, which is secreting juvenile hormone, right? And then we have another structure here that is known as prothoracic gland. So this is one that basically secretes the steroid hormone named as ecdysone. So ecdysone is falls under the group of hormones, group of steroid hormone named as ecdysteroid. And prothoracic gland secrete ecdysone, and ecdysone is controlled by a peptide hormone known as prothoracic gland hormone. Later on, we're going to look into that. Right. So in insect, the largest and most obvious endocrine glands are found in the prothorax here. It's called prothoracic gland. So it's basically the one that is producing a steroid hormone named as ecdysone or ecdysteroid. And it is also involved in molting of the cells, molting of the insects, right? So this is another, so you can see the front view of the insect's brain and the rear view of the insect's brain itself and how it is connected to a lot of other things. And from the rear view, we can see that there's a corpora cardiaca here and corpora alata. And these two are basically connected to the brain. Just like how you have your pituitary glands, right? It is connected to the brain. It's the same thing in insects. So we're going to look into prothoracic gland. It manufactures a steroid hormone known as ecdysteroid. It's a group of closely related steroid hormone, including ecdysone. So ecdysone is the most studied one. It stimulates synthesis of chitin and protein in epidermal cells, and it triggers a cascade of physiological events that eventually leads to molting in stages, right? In larvae stage, in pupa stage, ecdysteroid basically plays a role in driving the metamorphosis of insects. So because of that, ecdysteroid is also known as molting hormone. So once an insect reaches the adult stage, its prothoracic gland atrophy, it withers away and it will never molt again. So once the insect has already become an adult, it doesn't need to do molting anymore. So the prothoracic gland will then dies off and we no longer produce ecdysteroid. So what is molting? So you have heard me mentioning about molting before. So molting is basically shedding of exoskeleton of the previous life stage. What does it mean? It means that shedding of the exoskeleton of the old exoskeleton in order for the insects to grow from one stage to another, right? So without molting, an insect cannot grow. So you can try and recall back the video that we watched about butterfly, all right? The life cycle of a butterfly and how it becomes butterfly from a pupae, from a larvae stage. It involves many steps of molting process, and this molting process is basically controlled by hormone, mostly what we call as ecdysteroid hormone. So without molting, an insect cannot grow. So once a new critically fully formed an insect engages in species specific stereotype patterns of movement, molting behavior that liberated from the cuticle of the previous stage. So the timing of a molt is critical for two important reasons. First, the molt will fail and the insect will die if it is attempted prior to complete deposition of a new critical. So basically, molting process, it involves growing of the insect itself. And if you can still remember the video just now, in order for the insects to grow, it has to feed on fats, feed on lipids in order for them to grow in size, right? So each molting process will also involve will also result in growing of the insect itself, right? Second, the fresh cuticle of a newly molted insect is pale and soft because the chemical reaction that result in hardening and darkening take hours to days to be completed. So usually insects that undergo molting, they would go into seclusion, all right, because they are more vulnerable to predators, all right, and they would during molting process, what really happened is basically the replacement of the old cuticle into a new cuticle. And that new cuticle usually is pale and soft. It's not as strong as the old skeleton or the old cuticle. So it also involves other hormones to sort of sort of harden and darken this cuticle. So the hormones involved later on is going to be known as muscicon hormone, right? Later on, we're going to look into that. So this is the molting process. I see whether we can play this video or not of a tarantula, right? You can see that the new exoskeleton or the new cuticle, it seems fresh. It looks fresh. It looks pale. It's not as strong as the old cuticle one. And it would take hours, days for the new exoskeleton or new cuticle to become hardened, to become strong, right? And you can see at that stage, these tarantula is basically most vulnerable to predators. So that's why they always secrete themselves in order to do molting. So this is the exoskeleton. It's made up of chitin. Chitin is a long chain polymer of N-acetylglucosamine. It's a derivative of glucose. It is the primary component of the exoskeleton of arthropods, such as crustacean and also insects. If you guys still remember your biomolecule where we learn about monosaccharides, and one of the monosaccharide is glucose, and we learn also about monosaccharide derivative. So the exoskeleton of an insect is made up of a chitin. Chitin is basically a, I would say, a polymer, right, made up of glucose derivative named as an acetylglucosamine. So let me see whether I can show you guys the structure of an. Yeah, this is how it looks like. So this is how it looks like. This is a chitin. So it consists of a polysaccharide derivative, all right, known as N-acetylglucosamine. You can see here it's basically from a glucose derivative, and instead of OH, it bears the it bears the amine group and acetyl here, right? So basically, the exoskeleton of an insect is made up of chitin. Chitin is very hard, all right? It's very strong because it its main function is to basically envelop the organs in the insect itself, protect the organs inside the insects, right? So this is a prothoracic gland. So we have already established the understanding that prothoracic gland is involved in production of a hormone named as aglysteroid or aglycin. And prothoracic gland is basically controlled by um, It's basically controlled by a peptide hormone named as prothoracicotropic hormone or PTTH, and it is produced by copra cardiaca. Right? The copra cardiaca releases their stored PTTH only after they receive a signal from neurosecretory cells in the brain, act as a signal amplifier, sending out a big pulse of hormone to the body in response to a small message from the brain. This is what happened. This is the brain. And then when there is a need for the insects to move, so the brain will produce a brain hormone from the nucleosecretory cells. And then after that, it will then go to the copra cardiaca. Then this copra cardiaca will then produce a peptide hormone that basically called PTTH. And this PTTH will then travel to the what we call as prothoracic gland, asking the prothoracic gland to produce ecdysone. All right, so it's just like any steroid hormone that we have learned. So it needs to be stimulated by a peptide hormone. So its action is basically influenced by peptide hormone. In this case, ecdysone secretion is controlled by PTTH, and PTTH secretion is controlled by a brain hormone, all right, released from the neurosecretory cells in the brain. So this is copra alata. We have talked about copra alata a bit just now. So another pair of neurohumoral organs, it means that it is connected to the central nervous system by neurons and also by the blood vessel. So it lies behind the copra cardiaca itself. So we go back here. So we have copra cardiaca here and we have copra alata here for thoracic gland. And all of these structures are involved in producing hormones in insects, right? The copra alata, this function is to secrete juvenile hormone or JH, a hormone that inhibits development of adult characteristics during the immature stages and promotes sexual maturity during the adult stage. So juvenile hormone have two main functions. The first one is to inhibit development of of adult characteristics during the life cycles of an insect. So when the insect has already become an adult insect, it then have another function, which is to secrete or to stimulate the gonadal hormone, the sex hormone in insects. So it promotes sexual maturity during the adult stage. So neurosecretory cells in the brain regulate activity of the copra alata, stimulating them. So remember just now we have allostatin and allotropin, all right, stimulating them to produce JH during larvae or nymphal instars, inhibiting them during the transition to adulthood and reactivating them once the adult is ready for reproduction. So juvenile hormone, the the level is high during larvae stage, right? From one larvae to another when it wants to become from larvae stage 1 to larvae stage 2. This is when juvenile hormones level is high or concentration is high. And then when it starts to transition from pupa to adult, this is when the juvenile hormone level will go down. And when it becomes an adult, then it would then be secreted again in order to um, stimulate or activate the sexual or the sex hormone in insects. All right. So the neurosecretory cells in the brain is the one that control the production of copra alata. Okay, the cells produce and secrete brain hormone, a low molecular weight peptide that appears to be the same as or very similar to prothoracicotropic hormone manufactured by the copra cardiaca. So basically, we have copra alata, we have copra cardiaca. So it has different hormones that it synthesizes, and you can see that juvenile hormone is basically also a lipid-based hormone. Okay, it's made up from terpene. and it is controlled by the brain hormone that is being released by the neurosecretory cells in the brain, right? So GH secretion is controlled by allotropic and allostatin, and allotropic and allostatin are the one that are being released by the neurosecretory cells in the brain. So the main role of GH is in immature insects is to inhibit the genes that promote development of adult characteristic, causing the insect to remain as limb or larvae. So it depends on the life cycles of the insects whether it's going to become a limb or it's going to become a larvae. Uh, so we have two types of life cycles. Later on, we're going to look into that. Okay, so you can see here in adult insects, you can see that the brain release a brain hormone that reactivates the corpora allatum. So basically, juvenile hormone, like I mentioned just now, it has two functions. The first one is during development of immature insects, all right, in the form of limbs or in the form of larvae. And during this development of insects, the one that control the juvenile hormones are allotropin and allostatin, right? When it becomes an adult insect, the neurosecretory cells in the brain produce another hormone named as brain hormone. And this brain hormone will then go to the corpora allata and then it will stimulate the production of juvenile hormone. And what does juvenile hormone do to in adult insects? It stimulates yolk production, just like egg production in females and also in humans. So juvenile hormone, it stimulates yolk production in adult females and stimulate the male accessory glands to produce the protein of seminal fluids and spermatophore that also produce sperm under the influence of GH. So basically, adult insects um, produce juvenile hormone, and these juvenile hormones are the one that stimulate production of yolk in female and production of proteins and also proteins such as seminal fluids and also spermatophore, which basically will grow into a sperm, right? Because it's an adult insect, it's ready to mate. So juvenile hormone, like I told you guys just now, it's made up of a lipid-based structure. It's made up from a terpenoid structure, also a derivative of a lipid, and is the methyl ester of epoxyphenolic acid. So this cis-quiterpene exists in at least 6 different forms. You can have a look at it, and this is how it looks like. This is a juvenile hormone, right? So we have many types of juvenile hormone. This is one example, juvenile hormone 3, and this is how it looks like. It consists of lipid structure joined together. This is what we call as terpene, and you can see that there is also epoxy here, right? Epoxy methyl group, and this is how it looks like. And this is the excision. You can see the structure has a distinct penantine ring. So we have a cyclohexane here and cyclopentane here. And this is also a distinct structure of a cholesterol itself, a steroid. And this is how it looks like. Right? And this is just to show you an adult insects here. You can see there are ovaries that produce yolk production. And you can see that there's a brain there, corpora cardiaca just next to the brain. And you can see it's connected by the neurons, it's connected by the blood vessel. And right down the corpora cardiaca, we can see corpora allata here. And we can see prothoracic gland. And you can tell that all of these are basically connected to each other. And this one here is Drosophila, which is a fly, a house fly. This is how it looks like. This is in its larvae stage, pupa stage, all right. You can see that it has all these imaginal discs, right? And then when it becomes an adult, these imaginal discs will then transform into wings, right, into legs. So an imaginal disc is a sac-like epithelial structure found inside the larvae of insects that undergo metamorphosis. So once the larvae turns into a pupa, almost all the larvae tissue degenerate and the imaginal disc turn into the external structure of the head, right? You can see here. Um, and then it will then become thorax, limb, and also genitalia, right? So imaginaries are basically epithelial structures characteristic of insects that undergo metamorphosis, right? It transforms into um, different structures in adult insects. So the imaginaries differentiate into most of the structures of the adult insects such as legs, wings, and head. So the development of the imaginaries has been studied mostly in dipteran, this one here, Drosophila melanogaster, a model organism in the analysis of genetic and cellular regulation of animal development. So this one here, all these structures are the one that we call imaginal disc. So when it metamorphosizes into an adult insect, it would become other organs, right? It would become other structures like the eye, and then the legs, and then the wings, right? And genital apparatus is still there. When it is an adult insect, it becomes a mature genital apparatus that can eventually produce sperm, eventually produce eggs, right? In order for the insects to mate with each other and populate, right? So many other tissues and organs of the insect's body also produces hormone. So we have ovaries and testes, for example, produce gonadal hormones that have been shown to coordinate courtship and mating behavior. And we also have other structure known as ventral ganglia. So ventral ganglia produce two hormones here. We have eclosion hormone. Eclosion hormone is involved in egg disease process. Egg disease is basically the shedding of the old skeleton from the new skeleton when the insects undergo molting process. And then we have bursicon here. So bursicon hormone is the one that harden and darken the new exoskeleton, making it a strong exoskeleton for the insect itself. So insects also have other hormones, but it's not widely studied. And these hormones are worth mentioning in terms of it controls the level of metabolism in the insect, in terms of sugar, dissolve in the blood, adjustment of the salt and water balance, and regulate protein metabolism. So there's not a lot of studies on metabolism of insects, but we look more into the reproduction system, development system in the life cycle of an insect. So this one here might be of interest for the entomologists, all right? We're not going to go into that. So this is the second term that you guys have to know. So the first one just now is the molting process or molt. What is molting? And then the second one is metamorphosis. So metamorphosis is a biological process which an insect physically develops after hatching, involving a conspicuous and relatively abrupt change in the insect's body structure. So metamorphosis consists of irreversible changes in morphology, physiology, and behavior that takes place between the final larvae instar and emergence, the later being the molt that produces the adult insects, whether we have figure or not. So this one here is where the metamorphosis occurs. Yep, this one from larvae into pupa and from pupa into adult stage. So metamorphosis is necessary for exoskeleton limited growth. So metamorphosis, you can see that it is a complex and tightly regulated process that concludes larvae growth and result in the transformation to adult stage. So metamorphosis is basically the life cycle of insect cell. Since growth only occurs in the immature stages, adult body size is determined by the size the larvae attend when it stopped feeding and initiated metamorphosis. So metamorphosis is the step to grow for the insects to grow into adult insects. So metamorphosis is necessary for exoskeleton limited growth but permits partitioning of habitats and resources among life stages, right? So we have many types of insects. Later on, we're going to look into that. So this one here is an example of a metamorphosis in uh, common niche, right? Development is holometabolous with complete metamorphosis. The life cycle comprises distinct stages or instar separated by molds. A typical life cycle consists of an egg, at least 3 larvae instar, a pupa, and then an adult. So you can see here, this is the life cycle of a midge. And from there, you can see it has different instar of larvae. First instar, second instar, third instar, and fourth instar. And in order for the insects to grow from one instar to another, it involves molting process. This one here, all right, molting process. And we have apolysis, which is basically formation of a new exoskeleton, and ecdysis is shedding of the old exoskeleton. You can see here every time it happens, every time it transforms itself from one larvae to another larvae, okay, it will molt, right? And molting process involve these two processes, apolysis and ecdysis. And we can see that apolysis and ecdysis is also controlled by hormones such as juvenile hormone, ecdysteroid, and later on we can also see that there's also involvement of other hormones. And you can see here, this is what happened, metamorphosis from pupa to adult itself, and this is the adult stage, right? So we have insects that undergo this kind of life cycle called holometabolous. It's a complete metamorphosis. It has distinct stages of life, all right? Distinct stages named as larvae or named as instar. So from egg, it become larvae, and from larvae, it become pupa, and from pupa, it become adult. And we have another type of metamorphosis, which is called as hemimetabolous. So hemimetabolous is in complete metamorphosis, right? And the life cycle, the stages of the life cycle is called NIM. The holometabolous, holometabolous is called instar, and hemimetabolous is called NIMS. But it also involves ecdysis, it also involves apolysis, okay? So let's have a look, right? So we have 3 types of insects. So we have hemimetabolous, development or metamorphosis, and then we have hemimetabolous development. You can see that we have NIM, right, as instar, and then the adult insects would grow just like the NIM, would look just like the NIM structure, right? It's just that the adult has a more advanced structures in terms of it has a bigger in size, it has wings, you can see here. The juvenile forms closely resemble adult but are smaller and lack adult features such as wing and genitalia. The size and morphological differences between nymphs in different instars are small, often just differences in body proportion and the number of segments in later instar, so external wings part form. So we have 3 types of metamorphosis in insects. We have ametabolous, we have hemimetabolous, and holometabolism. Holometabolism is what we call as complete metamorphosis, and this one is incomplete metamorphosis. You can see the different. Complete metamorphosis, it means that it's different from larvae and it becomes pupa. Then you have a different structure when it becomes an adult. So you can compare it with the hemimetabolism. This one is incomplete metamorphosis. It means that the juvenile stage, the NIM stage, is basically just the same as an adult stage, the same in terms of the appearance of it. All right, it doesn't become completely different just like the one in holometabolism development. And you can see that the difference between the adult and the juvenile insects is only the genitalia maturation and also the wings production or the wings appearance in the adult insects. Right, so this is the description of it. Right, so our metabolism is basically characterized by no significant metamorphosis. We don't have significant metamorphosis. And as you can see, that's just a growth in size, right? And it's just the same structure between starting from the pronym until the adult insects. And hemimetabolism, the immature form resemble the adult form. And then holometabolism, it's a totally different thing. We have larvae, we have pupa, and we have adult in the form of insects. And this is basically the comparison between complete metamorphosis and incomplete metamorphosis. What are the types of insects that we have? So complete metamorphosis, it basically consists of 4 stages. We have eggs, larvae, and larvae we have different insta, and then we have pupa and also adult. For holometabolism or hemimetabolism, we have 3 stages: eggs and nymphs. Nymph have different also insta and also adult. Okay, so these are example of it: wasps, ants, fleas, and this one, and also we can say butterfly. This one happens in cockroaches, right? So when immature insects has grown sufficiently to require a larger exoskeleton, sensory input from the body activates certain neurosecretory cells in the brain, and these neurons respond, and it then triggers the corpora cardiaca to produce a peptide hormone named as PTH, and this PTH will then influence or induce prothoracic gland to produce adrenaline. Right. Let me see. So I understand that you guys have lecture at 10, other class, right?
Recording stopped.
Yes. So Peter, do you guys have class after this? Okay, all right. So at 11. So can I just proceed? Can I just proceed, Sofita, until maybe 10:30 maybe? Is it okay with you guys? Or you want, yeah. Yeah. Yeah, what about others? Are you okay if I proceed this until 10:30, those who have presentation? So no one is answering, so I guess it's okay. So I'm just going to proceed, and I'm just going to proceed with recording also.
Recording in progress.
Okay. So, all right. So we're going to look into hormonal control of molting and metamorphosis. So in the first 1 hour just now, we talk about the structures of the insect itself and what are the structures involved in terms of producing hormones. We have looked into the brain, we have looked into the corpora cardiaca and corpora alata, and we have also looked into prothoracic glands, and we have identified what are the hormones produced by all this year. And now we are going to look into the control, the regulation of the hormone itself in controlling the life cycles of an insect, right? So this is again an actin structure, how it looks like. It's a steroid-based hormone, right? So this is what happened when an insect or when an immature insect um, signaling the insect itself so that it can grow. So what really happened? It when an insect acquire a larger exoskeleton in order to grow, it will tell the brain to produce brain hormone, and this brain hormone will then travel to corpora cardiaca, and corpora cardiaca will produce a peptide hormone named as PTTH, and PTTH is the one that induce the prothoracic gland to produce a molting hormone named as ecdysone. And this ecdysone here, it's called a molting hormone because it basically drives the molting process, right? So go back to the previous slide. What does it mean by molting process? It's the growth of the insects by producing new exoskeleton to remove the old exoskeleton. So molting occurs every insta in the larvae. All right. So at the final mode, the adult emerges. So molting hormone affects many cells, but its principal um, function is to stimulate a series of physiological events known as apolysis that leads to synthesis of a new skeleton. So what apolysis mean? It means that the separation of the epidermal cells from the cuticle marks the beginning of the mold and the next insta. So apolysis is separation of old cuticle from underlying epidermal cells caused by release of active steroid at the beginning of each mold. So this is what we call as apolysis, the process of synthesizing new cuticle and then separate it from the old cuticle, right? So during apolysis, a new exoskeleton forms as a soft wrinkle layer underneath the hard part, exocuticle plus epicuticle of the old exoskeleton. So basically, during the molting process itself, we have um, a process known as apolysis, a production of a new exoskeleton. And when apolysis happens, the new exoskeleton will grow on top of the old um, exoskeleton. This is apolysis. And during apolysis, it can either take days or 2 weeks. Just now we have watched a tarantula do apolysis, right? And depending on the species and its characteristic growth rate. So once new exoskeleton has been formed, the insect is ready to shed what's left of its old exoskeleton. And this process is what we call as ecdysis. So ecdysis is basically shedding of the old skeleton from the new skeleton itself. So when insects have both old exoskeleton and new exoskeleton, at this stage, the insect is said to be parrot, meaning that the body is covered by two layers of exoskeleton, the old one and the new one. So molting or mold period during which the insect synthesizes new cuticle and other structures appropriate for next developmental stage begins with ecdysteroid-induced apolysis, ends with ecdysis of all cuticle. So this arrow just to show you the metamorphosis process, alright? So as long as ecdysteroid levels remain above a critical threshold in the hemolymph, other endocrine structure remain inactive, right? So end of apolysis, we did exclusion hormone to exclude the um, old skeleton from the new skeleton. Okay, I'll just show you this one here. All right, so this is the life cycle of an insect, particularly the butterfly itself. All right, so you can see the larvae stage and the pupa stage here, and eventually it would become an adult butterfly. So during the process of the larvae stage, so this is where the molting process occur. All right, so this molting process is production of new exoskeleton, and you can see that there is an increase in the aggresin level. So aggresin will drive a reaction known as apolysis, production of a new exoskeleton, and it can take days, it can also take hours, all right? And you can see when the new exoskeleton that have been formed, you can say that the level of eclosion hormone starts to increase. So eclosion hormone is the one that drives egg disease process, which is shedding of the old skeleton from the new skeleton. And when that happens, the new skeleton is very fragile, all right, and it needs another hormone named as bursicon hormone, also being produced by the ventral ganglia to harden and to thicken the new exoskeleton. Right? You can see here how does it do that? It's by tanning of the new exoskeleton due to the formation of quinone cross linkages in the exocuticle. Right? So this is the hormonal level of it. So in immature insects, juvenile hormone is secreted by the copra alata before each molt. Later on, we're going to have a look at it. So juvenile hormone, it inhibits the growth of the immature insect so that it won't become an adult insect before its time is up, right? It inhibits structures such as wings, reproductive organ, and external genitalia to mature before it's the right time, causing the insects to remain immature in the state of nymphs or in the state of larvae. So juvenile hormone is secreted during the larvae insta, during the nymph insta. And copra alata shrinks during the last larvae or nymphal insta and stop producing juvenile hormone. And this releases inhibition on development of adult structure and causes the insect to molt into an adult. So we're going to have a look into that. Uh, so this one here, all right? So this is an example of how hormones are being controlled in the metamorphosis or life cycle of an insect itself. This one is in butterfly. And you can see it start off with larvae stage. In the larvae stage, you can see that the juvenile hormone secretion is very high, and you can have the aggresin hormone is also high every time it tries to move here. And you can see ECH is eclosion hormone, which drive the egg disease formation. And you can see here the PTTH hormone um, also increases, and this PTTH hormone increase leads to the increase of aggresin hormone, which basically drive the molting process, right? And every larvae stage, you can see it's the same regulation of hormone here. We have juvenile hormone, we have aggresin, we have PTTH, and at the end of the molting process, we can see there's a surge in eclosion hormone, which drive the egg disease process. And you can see here from larvae stage to pupa stage, there's no juvenile hormone being produced, only PTTH and only aggresin, right? Because the insects is starting to grow into an adult insect itself, right? So you can see that there's no juvenile hormone being produced in the pupa stage and in from pupa to butterfly stage, right? And you can see that when it becomes an adult, the brain hormone will be released, and that brain hormone will then induce the production of the juvenile hormone from the corpora allata, and this juvenile hormone will then induce the maturation of the gonads hormone, gonads structure in the adult insects, right? So that's how the regulation happens. Okay. So this is another graph that shows you how the regulation of the hormone occurs. You can see here, uh, this is the time taken for the life cycle of an insect, the molting process. The molting process involves first the secretion of the brain hormone, and this brain hormone will then go to the corpora cardiaca, and this corpora cardiaca will produce a peptide hormone named as PTTH, right? And this PTTH will then go to the prothoracic gland and ask the prothoracic gland to produce ecdysone or ecdysteroid. And you can see that's an increase in ecdysteroid every time the insects molt. And then at the end of the molting process or apolysis process, you can see that there's another hormone that the level is high, we call as eclosion hormone. And the last hormone here is when the shedding of the old exoskeleton finishes, then it will then produce another hormone that is known as a bucicon hormone to darken and harden the new exoskeleton, right? Okay, this one here has already um, been explained just before this. So you can have a look at this one here. But one thing that I want you to take note is in the adult production of juvenile hormone is resumed, but now the hormone acts as a gonadotropic hormone, stimulating the development of the gonads. So juvenile hormone have two functions, all right? So the first one is to keep the insects from maturing, and the second one in adult insect is to induce gonadotropic development or gonad development. So this is a table here that shows you the hormone, the source or chemical nature, the function itself. So we have ecdysone produced by the prothoracic gland. It's a steroid base and it stimulates molting. And we have juvenile hormone. It's excreted by corpora allata. It's an isoprenoid or terpene structure, and it directs metamorphosis. And then we have PTTH being produced by the corpora cardiaca, right? It's a peptide hormone. It stimulates ecdysone. And we have allatotropin, allatohibin, produced by the neurosecretory cells in the brain that controls the juvenile hormone. And we have eclosion hormone and vesicant hormone, both produced by the ventral ganglia itself. Um, Eclosion stimulate eclosion, or it also can be called as egg disease process. And then we have bursicon here. It stimulate tanning, it stimulate hardening of the new exoskeleton. And this is another figure to show you the control of the hormone itself. You can see larvae to larvae insta. You can see the ecdysone level is high. Juvenile hormone is also high, and ecdysone is produced by prothoracic gland. And you can see PTTH is the one that is being produced by the corpora cardiaca that induce the production of ecdysone from prothoracic gland. And ecdysone is being produced in the larvae stage, larvae to larvae stage, larvae to pupal stage, and pupal to adult stage. And you can see that corpora allata produces juvenile hormone at high concentration through the development of larvae insta, and very low concentration during larvae to pupal, and there's no production of JH when pupa is being converted into adult or is being metamorphosized into adult. So I've already explained this one here. So the role of ecdysone in metamorphosis has been intensively investigated. So ecdysone is present prior to the beginning of metamorphosis. So in the larvae stage, Um, each mode is triggered by pulses of egg descent, but the presence of juvenile hormone prevents the larvae from forming a pupa. You can see here, during the larvae stage, egg descent level is high and the juvenile hormone level is also high. But when larvae wants to become a pupae, you can see that there's no production of juvenile hormone. There's only production of ecdysone, and there's only production of ECH and PTTH. And from pupa to butterfly, or pupa to adult butterfly, you can see there's no more production of what we call as juvenile hormone. And juvenile hormone starts to being produced again in adult insects in order to induce gonadotropic function. Production of egg or production of yolk in female, and production of seminal fluid, and also spermatophore in adult insects, in male insects. One here. So in the absence of normal juvenile hormone production, the adult remains sexually sterile. So this is just to show you hemimetabolist development. This is in complete metamorphosis, eggs, and then it becomes nymphs. We have different instar of nymph, and adult it resemble the juvenile insects or juvenile stage. And holometabolist, it's a complete metamorphosis. It means that it's a complete different structure. It's a complete different appearance in insects. From egg to larvae, we have different instars of larvae, and then it become pupa. Then pupa will become adult. So 4 stages of development. And hemimetabolist, 3 stages of development. And there's also a study that has been done um, that basically shows what's going to happen if copra alata is being removed, or if copra alata is being implanted. You can see here, right? So this is a normal adult. This one here, the B, is a normal insect development or normal life cycle of what we call as holometabolous insects. So from egg, and then it become larvae. So each stage or each instar of the larvae itself, it requires molting process, and you can see that the steroid would be high, the juvenile hormone would also be high, accompanied by PTTH hormone. And at the end of the molting process, you can see that's also a high level of eclosion hormone, and it will then become the late stage of the larvae itself. Then will molt and become a pupa. And this one here, it only involves the uh, role of hormones such as PTTH, and also steroid, and also eclosion. There's no involvement of juvenile hormone because the insects is in the way to be an adult insects. So it doesn't really need to be the juvenile hormone itself. And from pupa, it become an adult. It molts again and become an adult. So what happens if there is no copra alata in an insect? Copra alata is being removed, so there's no molting process, right? So copra alata produces juvenile hormone. So juvenile hormone is needed for development of larvae stage. So every time the larvae molt, it would also require juvenile hormone. So when there's no copra alata, juvenile hormone is not being released. Instead of going through multiple instars of larvae, the larvae would molt straight into pupa. And from there, the insects won't have like a sufficient growth. And that's why the adult insect would become small in size. And what happens if we have active copra alata implanted in the insect itself? In the final stage of the larvae, you can see that the larvae it has like a high amount of juvenile hormone. So when it have a high amount of juvenile hormone, it will continue growing in the larvae stage until it becomes big. And you can see that it results in a big pupa, and it results in a big adult insects formation. And from there, we can say that we can manipulate the growth of an insect by manipulating the copra alata, uh, and also by manipulating the juvenile hormone level in insects. And it's the same experiment here, right? So copra alata removed in the third insta, you can see a small adult insect is being produced. And the normal insect, this is how it looks like. So we can manipulate insect hormone by manipulating the juvenile hormone action. And juvenile hormone action is a potential target of pest management. So basically, we can either inhibit the juvenile hormone in the insects in order to kill it at early larvae stage, or we can also knock down the juvenile hormone or remove the copra cardiaca, um, in remove the copra alata in the insects or in the larvae itself. So we can also have enzymes such as juvenile hormone epoxy hydrolase. It basically hydrolyzes the juvenile hormone so that the juvenile hormone won't be functional, won't be there in the insects, and the insects won't grow into larvae stage, and it would become smaller in terms of size. You can see here, juvenile hormone epoxide hydrolase has attracted great interest because of its critical role in the regulation of juvenile hormone in insects. In this study, one JHEH gene from Apologus lucarum was characterized in terms of reduced amino acid sequence, phylogeny, homology modeling, and docking stimulation. So the result revealed a conserved catalytic mechanism towards JH. So basically, the gene here from Apologus lucarum can control or can inhibit the juvenile hormone. This is like an abstract from one of the journal articles. You can see here. Because GH is a specific hormone in insects which plays important roles in the regulation of physiological processes in development and reproductive maturation, interfering with its biosynthesis or degradation by juvenile hormone epoxide hydrolase, can result in a promising strategy for alternative insecticide with low toxicity to non-target organism. So juvenile hormone is very specific to one insect. So if we target that insect, it won't be um, dangerous to other non-target organisms. So most of the research on JH degradation has been concentrated on the mechanism of action of an enzyme known as esterase or juvenile hormone esterase or juvenile hormone epoxy hydrolase. Is the one that degrades the JH. So these enzymes here can be mixed together in a liquid and then can be applied to insects, and this would then inhibit the JH by degrading the JH. So a characterization of JHEH in the green plant bark is important because of its role in JH regulation and the irreversible degradation. So what they did, they basically tried to knock down the genes in insects, or they can also isolate this enzyme here and then apply it to insects in order to um, manage pests in agriculture, right? So that's about it for the insects hormone. So before I dismiss the class, I'm just gonna recap back what we have learned for the last 1.5 hours in terms of the insects hormone itself, right? Let me just open up my my OneNote, right? So you try and go back to the learning outcome itself. What does it tell you about learning outcome and whether you have achieved all that learning outcomes from today's lecture? So we learn about insects hormone, right? So first of all, we learn about the structures in the insect itself. What are the structures involved to produce the hormone? In hormone production, right? So we have looked into the brain itself that contains the neurosecretory cells, and then we look into the corpora cardiaca. So it produces what we call as PTTH, and then we have right after that corpora alata. So this one here is producing ecdysteroid or ecdysone, and then we have prothoracic gland. This one here is not ecdysteroid, but instead juvenile hormone. Right, so this one here is ecdysteroid. Okay. And you guys have to know what is molting and what is metamorphosis, right? So molting is basically the growing of the cells itself, the growing of the insects, right? By a process known as apolysis, formation of new exoskeleton. Right? And then we have metamorphosis. Okay. And we have looked into the life cycle of the insect itself. The one that is important is hemimetabolis. And then we have holometabolis. So one is incomplete metamorphosis, another one is complete metamorphosis. So egg, nymph, adult. That is the first one. And then we have egg, larvae, pupa, and also adult. So you have to know what are the differences between these two, right? So adult insects resemble the juvenile or immature insect. And this life cycle of the cells or of the insects are controlled by hormones, right? So we have hormones that drive the molting process known as ecdysone or ecdysteroid, and larvae to larvae. We have juvenile hormone is also high, and then we also have other hormone PTTH, and at the end of the molting process, we have another hormone known as eclosion and persicon. Right. Okay, these are all the four hormones that are important. And juvenile hormone, it is controlled by allostatin from the brain. Okay, so this one here is during larvae and larvae stage. So it inhibits, JH inhibit maturation of insects. So that's why JH level in pupa to adult stage, right? JH level is very low, low or none. Okay. And JH also have another function in adult, what we call as gonadotropin function. So yolk sac in female, and then seminal fluid and spermatophore, right? You can check the spelling in male. So this would then allow mating of insects. Okay. And then we have also looked into manipulation of JH. So you can refer back to the notes that I showed guys about the manipulation. Okay. Um,
Recording stopped.
all right. That's what we have learned in insects hormone. Any question from you guys before
Uh,
we end the class?
Doctor,
Yes.
you just said that the juvenile hormone is inhibit the maturation uh, for the insect, but it is required for the growth of the pupa. So it is not. I don't understand that part.
It is required for the growth of pupa. Did I say that?
It
Right.
is like
Yeah, I don't think I said that, but you can refer back to your lecture note. Okay. You can refer to this one here. Right. So this one here, I told you guys that it JH hormone. It basically is
doctor.
secreted in the very
Are you
yes,
showing something?
yes. Can you see it now?
Yes.
All right. So this one here. Okay, let me just. All right. So we have JH hormone here. Um, so JH hormone. So we have the first one, search, and the second one, and there's no JH hormone here. No JH hormone. Okay, no JH. So it only is required during larvae stage. Larvae to larvae stage. So from one instar to another. But it's not required from larvae to pupa stage.
So the larvae to larvae stage, they inhibit maturation.
Yes, because it's in larvae stage. So it's still a larvae. It just grow in terms of size, but it doesn't grow in terms of it doesn't have any other structures like wings, things like that.
So
So again,
is it maturation and growth different?
yes, right. So it only involve like growth in size, grow in size. So if you watch, say, remember the video that I gave you guys there, the one that we watched earlier. So larvae, in order for larvae to grow, it requires like feeding on fat, right, as food, and it grows in terms of size. But um, the larvae stage, the pupa stage, this is a different stage. So pupa is like one step closer to become an adult. And in pupa, this is where we have imaginal yeast that eventually will develop into organs in adult insects, right? So development into adult insect, it doesn't require juvenile hormone. It's called a juvenile hormone because it keeps the insects to be juvenile. Does it make sense?
Yes. Thank you,
All
doctor.
right. Okay. Any other questions? Right. calling once, calling twice, and calling thrice. If there is no question, before I dismiss the class, all right. So for those of you who haven't filled up the teaching assessment form, do fill it up because do fill it up, all right, before I put the second test marks this weekend. And I would also fill in your attendance in ESMP. So you can check your attendance on ESMP by this weekend. And if you don't have more than 80% of attendance, unfortunately, you cannot go into the final exam. You cannot sit for the final exam. I won't accept any excuses unless you have a a letter from a certified GP, say for example, saying that you have problems with your health. Uh, so I would consider you to sit for the final exam. If you don't have enough in terms of 80% of attendance, you won't be allowed to sit for final exam. So that's all from me. So we have another one final topic. So as agreed by you through Sofita, we'll do the final topics in 2 days. Within in 2 days during the study week. All right. So we're going to talk about plant hormone itself. Okay. Um, if there is any question that you guys have after this, you can definitely relay the message to Sofita or you can just email me. All right. So that is all from me. Class is dismissed. I guess I see you next week then.
Thank you, doctor.
Right.